Now we are going to learn about genetic transcription of the genetic code. Transcription is the transfer of stored information from DNA to RNA in the form of nucleotide sequence. So it is accomplished by DNA-dependent RNA synthesis using DNA strand as template. In this process, an mRNA copy of a DNA sequence is produced as the first step of gene expression, making use of the base sequence of one DNA strand, termed template strand or antisense strand. The whole process is mediated by RNA polymerase. Next, transcriptional units, transcriptons. The elementary unit of transcription is known as transcripton. It is a small portion of a DNA molecule where actual transcription occurs. Only one strand of the transcripton is transcribed. It is known as the template strand, antisense strand, or anti-coding strand. The non-transcribing strand is called the non-template strand, sense strand, or coding strand. The same strand of a DNA duplex may serve as the sense strand in one transcription and as the antisense strand in another. The RNA transcribed from an antisense strand is known as RNA transcript. It might be an exact replica or negative copy of the antisense strand and an identical copy of the sense strand, except that thymine is replaced by uracil. RNA transcript is synthesized by the polymerization of ribonucleotide phosphatase like ATP, UTP, GTP, and CTP. During this, the 3' OH of one nucleotide reacts with the 5' OH of another, forming a phosphodiester bond. The sequence in which ribonucleotides are added to the growing RNA chain is determined by the sequence of the bases in the template strand. New nucleotides are added to the 3' end of the growing chain. This transcription always occurs in the 5' to 3' direction. Transcriptions occurring regions like transcriptons have some functionally different regions. In prokaryotes, they include the leader, promoter, operator, cistronic, terminator, and trailer regions. In eukaryotes, they include the leader, promoter, acceptor or regulator, cistronic, terminator, and trailer regions. Cistronic region consists of only exons and prokaryotes, but in eukaryotes it has alternating exons and introns. Only exons code for amino acids, so they are transcribed, translated, and genetically expressed. On the other hand, all other sequences are non-coding, so they are only transcribed but not translated and genetically expressed. When you look at the prokaryotic transcripton structure, we could see regions like leader, promoter, operator, cistrons, terminator, and trailer. But eukaryotic transcripton consists of leader, promoter, acceptor, cistron, terminator, and trailer. Leader region plays some role in determining the rate of transcription. Promoter region provides the recognition signal to initiate transcription. In most eukaryotic RNA polymerase II, it contains the CAT box sequence and the TATA box sequence. The former serves as a recognition site for RNA polymerase and the latter provides a binding site for the enzyme. The binding of RNA polymerase to the TATA sequence initiates transcription, and acceptor or prokaryotic operator region serves as the binding site of the regulator which influence transcription. The exons of the cistronic region contain the structural genes which code for amino acids and thereby provide the information for transcription. Terminator region provides the halt signal for the transamination of transcription. Trailer is a non-translated sequence following the terminator codon. Cistronic region is monocistronic in eukaryotes but polycistronic in prokaryotes. Then transcription enzymes. RNA synthesis is mediated by the enzyme RNA synthetase or RNA polymerase. It organizes and recognizes an appropriate promoter site in the antisense strand of the DNA transcription and binds with it. Then it mediates the synthesis of RNA using the antisense strand as a template. So in prokaryotes, there are two kinds of RNA polymerases. One of them produces the RNA primer during DNA replication and the other one mediates the transcription of all the three kinds of RNA like mRNA, tRNA, and rRNA. In eukaryotes, there are three kinds of nuclear RNA polymerases in addition to the mitochondrial and chloroplast RNA polymerases. The three classes of nuclear RNA polymerases are polymerase 1, 2, and 3. The first one is located in the nucleolus and the others are localized in nucleoplasm. Polymerase 1 governs the transcription of all rRNAs rather than 5S RNA. Polymerase 2 mediates the transcription of mRNA and some nuclear RNA, and polymerase 3 is involved in the transcription of tRNA, 5S RNA, and some other small RNAs. Then bacterial RNA polymerase is a hollow enzyme. It has two components, namely sigma factor and core enzyme. The core polymerase is composed of 5 polypeptide subunits, namely 2 identical alpha subunits, 1 beta subunit, and 1 beta bar subunit, and 1 omega subunit. Only the holoenzyme can initiate genetic transcription. The sigma factor would be released soon after initiation. Eukaryotic RNA polymerases are more complex. This is because there are about 10 kinds of subunits in them. The 3 classes of eukaryotic RNA polymerase, like 1, 2, and 3, differ from each other in their subunit composition. Some subunits are common for all of them, but the others are different. Mitochondrial and chloroplast RNA polymerase are very simple and they have only a single subunit each. Sigma factor has 3 major roles, namely it recognizes the polymerase recognition site of the promoter region of the DNA template, enables the binding of the enzyme to the binding site of the promoter, and initiates RNA synthesis. On the other hand, core enzyme is concerned with RNA polymerization, elongation of RNA chain. The catalytic site of RNA polymerase is believed to be located in the beta subunit. Once the RNA chain has begun polymerization, the sigma factor dissociates from the enzyme and the core enzyme comes into action for chain elongation. Then transcription factors, TFs. In addition to the RNA polymerase, some protein factors are also involved in mediating the transcription process. They are generally called transcription factors. They control the rate of transcription by binding to a specific DNA sequence around the TATA box. This binding forms a platform for the binding of RNA polymerase 2. Transcription factors are essential for the stable binding of RNA polymerase with the template strand of DNA, for the initiation of transcription at the correct codon, and also for the final release of RNA polymerase and RNA transcript from the template strand. Genes without TATA box may have an initiator element. Still, other genes have neither TATA box nor initiator element. Then components of transcriptional activity. Transcriptional activity is the rate of RNA synthesis, the number of full-length RNA transcripts synthesized per minute. Transcription initiation is usually rate limiting and also the primary level of gene regulation in prokaryotes and eukaryotes. So transcriptional activity directly reflects the efficiency of transcription initiation. The control of initiation involves 3 major components, namely basal components, constitutive components, and regulatory components. Basal components serve primarily to direct RNA polymerase to the start site of transcription, permitting the initiation of RNA synthesis. Then constitutive components control the efficiency of initiation in the absence of regulation and allow the expression of different genes at different rates to meet the basic needs of the cell. Regulatory components alter initiation efficiency, allowing the modulation of the transcriptional activity of individual genes in response to changes in the environment. Then transcription in prokaryotes. In prokaryotes, transcription is completed in 3 major stages, namely initiation, elongation, and termination. First, initiation of transcription involves all the molecular events occurring before the incorporation of first nucleotide. It is completed in 3 main steps as follows, like binding of the holoenzyme RNA polymerase with the promoter region of DNA, local unwinding of the companion strands of DNA. This is induced by RNA polymerase, which has both DNA unwinding and DNA rewinding powers. Local unwinding makes the template strand free for base pairing with free ribonucleotides. Then phosphodiester bonding between the newly incorporated initial ribonucleotides. During initiation, the sigma factor of RNA polymerase recognizes a specific sequence in the promoter region of the template strand known as recognition site. This may be called template recognition. Soon sigma factor complexes with the core enzyme and forms the RNA polymerase holoenzyme. The holoenzyme interacts with the binding site in the promoter region, then moves to the operator region, and finally comes to the initiator site in the cistronic region to initiate transcription. Transcription virtually begins at the initiation site. Factors influencing initiation, elongation, and termination of transcription, like transcription, promoter, operator, cistron, terminator regions are there. Then template recognition by sigma factor, core enzyme will be there, RNA polymerase or holoenzyme. Then alpha factor released RNA chain elongation, core enzyme released mRNA transcript formed. So there are different sites like recognition site, binding site, template strand, initiation site, termination codon. Sigma factor has a significant role. It recognizes the promoter region and ensures that RNA polymerase binds exactly with the binding site. It can bind with the binding site only when it is complexed with the core enzyme. The core enzyme cannot exactly recognize the promoter sequence all by itself, and so it cannot initiate transcription without complexing with the sigma factor. The binding of the sigma factor or RNA polymerase with the promoter region causes the local melting or disruption of hydrogen bonds and the unwinding of and separation of the companion strands of the DNA duplex. The unwound region of the transcription is sometimes called transcription bubble. It moves forward along the polymerase to avoid strain on the adjacent intact regions of the DNA double helix. Only a short region, 12 to 70 BP, is unwound at a time. Soon one strand of the bubble acts as a template strand and directs the synthesis of an mRNA on it. Once the mRNA chain has become a few nucleotides long, usually 2 to 10, a sigma factor is no longer required. So it detaches from the core enzyme and the DNA template and becomes free to get complexed with other core enzymes to form yet another holoenzyme. At the same time, the core enzyme comes into action, moves forward, and brings about chain elongation. So step elongation. Elongation or polymerization of mRNA chain occurs within the second step. Initiation happened, now elongation is happening. So elongation or polymerization of the mRNA chain occurs within the transcription bubble. Soon after the release of sigma subunit, it is catalyzed by the core enzyme of RNA polymerase, which keeps on moving along the template strand in the 3' to 5' direction. As the core enzyme moves forward, it unwinds the intact DNA strand ahead of it and at the same time rewinds the separate strands behind it, leaving the transcription bubble in between. Soon activated ribonucleotide phosphates like ATP, GTP, CTP, and UTP covalently bind to the corresponding complementary nucleotides of the template strand through hydrogen bonding. Adjacent nucleotides are then linked together through energy-rich phosphodiester bonds with the help of core enzyme. The energy required for this is made available by the hydrolysis of the terminal bonds of nucleotide phosphatase. Chain elongation always occurs in the 5' to 3' direction. The binding of RNA polymerase to DNA and the growing mRNA maintains the stability of the transcription complex. Then RNA polymerase has no proofreading powers for correcting mistakes and repairing damages. Therefore, the incorporation of ribonucleotides during chain elongation must be absolutely error-free. Otherwise, there is high probability for mismatch, binding, and copy errors. Then elongation of the mRNA chain initially forms a transient and very short mRNA, DNA-mRNA hybrid or heteroduplex. Soon the 5' end of the mRNA strand progressively separates from the DNA strand. As a result, the template DNA strand becomes free from the mRNA transcript and the two DNA strands reassociate to form the DNA duplex. Then termination. Termination of transcription occurs at a specific DNA sequence which serves as a stop signal from RNA polymerase. When the core enzyme of the RNA polymerase reaches this sequence, the transcription bubble collapses, the transcription complex dissociates, the core enzyme. So adjacent nucleotides are then linked together through energy-rich phosphodiester bonds with the help of core enzyme. The energy required for this is made available by the hydrolysis of terminal bonds of nucleotide phosphatase Then chain elongation always occur in the 5' to 3' direction. The binding of RNA polymerase to DNA and the growing mRNA maintains the stability of the transcription complex. Then RNA polymerase has no proofreading powers for correcting mistakes and repairing damages. Therefore, the incorporation of ribonucleotides during chain elongation must be absolutely error-free. Otherwise, there is high probability for mismatch binding and copy errors. Then elongation of the mRNA chain initially forms a transient and very short DNA-mRNA hybrid or heteroduplex. Soon the 5' end of the mRNA strand progressively separates from the DNA strand. As a result, this template DNA strand becomes free from the mRNA transcript and the two DNA strands reassociate to form DNA duplex. Then transamination. Termination of transcription occurs at a specific DNA sequence which serves as a stop signal for RNA polymerase. When the core enzyme of the RNA polymerase reaches its sequence, the transcription bubble collapses and the transcription complex dissociates. The core enzyme and the mRNA transcript are released and the DNA returns to its double helical state. Transcription termination involves three major events, namely cessation of polymerase movement and termination of chain elongation at the terminator sequence, then release of the mRNA transcript from the core enzyme and dissociation of the core enzyme from the DNA template. The released core enzyme will soon bind to a segment in the DNA and will move to the promoter for complexing with a sigma factor. Then types of termination. Based on the involvement of chain terminating protein called the Rho factor, two kinds of transcription termination can be recognized among prokaryotes, namely Rho independent and Rho dependent. The functional form of Rho is a hectamer with 6 identical copies. Rho independent termination can occur even if Rho is not present. On the other hand, Rho dependent termination occurs when Rho is present. In Rho independent termination, RNA polymerase continues to mediate transcription past the terminator sequence by a process called read-through. Then Rho independent termination. Rho independent termination is also called intrinsic termination. It is built-in mechanism in which the transcribed mRNA transcript causes and contributes to the termination of its own elongation. In this case, the stop signal is provided by a specific terminator sequence in the mRNA transcript without the participation of Rho factor. The core enzyme recognizes the signal sequence and stops moving beyond it. As a result, mRNA polymerization stops. But in some instances, there will be special transcription terminator sequences called attenuator sequences which may cause optimal termination. They permit transcription to continue when the gene product is insufficient and terminate transcription when there is enough gene product. Then Rho dependent termination. In Rho dependent termination, Rho factor plays an active role in it. First recognizes the stop codon in the complex template DNA. Then it binds with the core enzyme. When the enzyme reaches the terminator sequence, this Rho factor core enzyme complexing brings the enzyme to a halt and disrupts the base pairing between DNA template and RNA transcript, mRNA transcript. Also, it causes the detachment of the mRNA from the DNA and the enzymes and the release of core enzymes from template DNA. This in turn terminates transcription. The mRNA transcript releasing from the enzyme is known variously as heterogeneous nuclear RNA or hnRNA, primary mRNA, precursor mRNA or pre-mRNA. Then transcription termination through stem loop configuration. In bacteria, Rho independent and Rho dependent terminations of transcription occur at a specific sequence in the mRNA transcript known as inverted repeat sequence. In it, the first half of the sequence is followed by its inverted repeat sequence in the second half with a short sequence in between. Just before the last base is transcribed, the inverted repeat sequence assumes a hairpin-like stem loop configuration in the mRNA strand through intramolecular base pairing. The stem loop structure acts as a signal for termination. Also, it enables the detachment of mRNA transcript from the template strand of DNA. Now, we are going to learn about transcription in eukaryotes. Earlier, we have learned about transcription in case of prokaryotes. Next, we are going to learn about transcription in eukaryotes. Eukaryotic transcription is almost similar to that of prokaryotes. However, it differs from prokaryotic transcription in the following respects. There are 3 types of RNA polymerases, 1, 2 and 3. Only one type in prokaryotes. mRNA synthesis is catalyzed by RNA polymerase 2. There are 3 different types of promoters for the 3 classes of RNA polymerases. Only one type of promoter in prokaryotes. Initiation of transcription is more complex than in prokaryotes. Termination of transcription does not involve stem loop structure. Stem loop is present in prokaryotes. Binding of RNA polymerase 2 to its promoter involves a number of proteins called transcription factors. No transcription factors in prokaryotes. Then nearly 50% or more of the promoters contain a specific sequence, TATAAT, called Tata box or Goldberg-Hugeness box. Goldberg-Hugeness box. In some genes, other sequences such as CAAT box may be present beyond TATA box, absent in prokaryotes. They act as binding sites for the transcription factors which stimulate or repress transcription initiation. Many genes may contain sequences such as enhancers and the silencers located outside the promoter, absent in prokaryotes. Enhancers induce and silencers inhibit transcription. Termination of transcription in eukaryotes occurs at a point much beyond the end of the protein coding sequence. The actual mechanism of termination is not definitely understood. It is generally held that definite termination signals do not exist in eukaryotes. At the same time, dissociation of a transcription factor at the same point is believed to destabilize the transcription complex. This in turn causes the detachment of RNA polymerase 2 from the template strand, resulting in the termination of transcription. Then transcription complex. The major components of eukaryotic transcription complex include RNA polymerases, transcription factors, promoters, enhancers, and silencers. So RNA polymerase. In prokaryotes, there is only a single kind of RNA polymerase. But in eukaryotes, there are 3 classes of structurally and functionally different kinds of RNA polymerases, 1, 2, and 3. RNA polymerase 1 is located in nucleolus and it catalyzes the transcription of class 1 nuclear genes to synthesize all other types of rRNAs other than 5S RNA. RNA polymerase 2 is located in nucleoplasm and it governs the transcription of class 2 nuclear genes, which include all protein coding genes and some genes that code for small nuclear RNAs. RNA polymerase 3 is also located in nucleoplasm and it mediates the transcription of class 3 nuclear genes to synthesize tRNA and 5S RNA. Next, transcription initiation factors or TIFs. TIFs, also called basal transcription factors or BTFs, or general transcription factors or GTFs, are the accessory protein factors which assist RNA polymerases to recognize their promoter. They bind with the DNA to form a complex which recruits RNA polymerase to the transcription initiation site of the DNA. The TIF and the enzyme collectively form the pre-initiation complex which forms the basal apparatus of transcription initiation. Then eukaryotic promoters, enhancers, and silencers. Promoters, enhancers, and silencers are cis-acting regulatory sequences. They are capable of local interactions with the pre-transcription initiation complex in eukaryotes and bacteria. They interact with RNA polymerase. In eukaryotes, constitutive transcription regulators usually bind at sites upstream from the basal promoter. In bacteria, they flank the promoter. Enhancers and silencers are distant regulators located outside the promoter, hundreds or thousands of base pairs away from the genes they control. Enhancers are positive regulators which stimulate transcription, and silencers are negative regulators which inhibit transcription. They are quite common among eukaryotes but rare in prokaryotes. Then, Eukaryotic promoters. Promoters are cis-acting control sites like cis-acting sequence, next page, proximal to the genes they control. The promoter consists of two components, namely basal promoter and upstream promoter. RNA polymerase binds with the former or basal promoter. The later contains constitutive and regulatory enzymes or elements that collectively control transcription initiation. The promoter for RNA polymerase 1 is specific in the sense that the RNA polymerase of a particular species can bind only with the promoter of that species. The promoter of RNA polymerase 2 in most cases may contain TATA box and CAAT box. TATA box pronounced as Tata or Goldberg-Hagen's box. Tata box is a eukaryotic promoter element to which RNA polymerase binds during transcription. This is usually a 6 to 7 base pair long nucleotide sequence like TATA like that, located 90 to 27 base pair upstream from the start point of eukaryotic structural gene. TATA box is analogous to the Pribnow box of prokaryotic promoters. Pribnow box is a highly conserved prokaryotic promoter element to which the sigma subunit of RNA polymerase binds during transcription. It is a 6 or 7 BP long segment located nearly 10 BP upstream from the start point of transcription site. The commonest Pribnow sequence is TATA-AT or TATA-ATG. CAAT box. Bonoist et al. In 1980 is a common promoter sequence of many eukaryotic genes that are transcribed by RNA polymerase 2. It is found upstream of TATA box. It probably plays an important role in promoter function and is necessary for efficient initiation of transcription. Then eukaryotic enhancers. Enhancers are short cis-acting and distantly located positive gene regulators or positively acting control sites which can enhance transcription activity. Quite often they contain both constitutive and regulatory elements which can increase transcriptional activity up to 1000 times from a very great distance. Enhancers do not show promoter activity because they lack basal components. They may contain functionally clustered groups of binding sites termed enhance cons. Many transcription enhancers contain the conserved octameric sequence of ATTGCAT with which some transcription factors interact. Some enhancers are constitutively expressed in most types of cells, while the others are tissue-specific or species-specific. Enhancers act by increasing the number of RNA polymerase molecules. Their enhancer activity is mediated by sequence-specific RNA binding proteins. The attachment of DNA binding protein to an enhancer causes the looping out of the nucleotides between the enhancer and the promoter. This looping brings the enhancer into physical contact with the promoter of the gene it enhances. The loop structure then facilitates the binding of RNA polymerase with the transcribing gene. Then eukaryotic silencers. Silencer sequences are short, distantly located and cis-acting negative regulators or negative control sites. They are common in eukaryotes but rare in prokaryotes. In E. coli, they are being associated only with the genes that are transcribed under the control of delta 54 factors. Silencers may be located upstream or downstream of the genes or rarely within the genes they regulate. They can bind with constitutive and regulatory transcription factors and interact with the basal interaction complex to control transcription initiation. Then silencers exert a negative effect on the rate of genetic transcription. They repress promoter activity in those cell types rather than target cells. So they are cell type specific negative transcription regulators, or in brief, cell type specific transcription inhibitors. Then cis-acting DNA sequences. Cis-acting elements are short eukaryotic DNA nucleotide sequences which can regulate the genes that are seen on the same DNA or chromosomes on which they are located. The gene synthesis configuration without any effect on the genes on other DNA or chromosome. Genes in the trans configuration. A cis acting element usually consists of 10 to 12 BP and is located either within the gene or within the 5' flanking region. Cis acting sequences represent the recognition sites in which specific recognition factors interact. They are essential for eukaryotic gene regulation. In general, they do not code for proteins but rather serve as attachment sites for DNA binding proteins. Enhancers, silencers, promoters, and operators are examples of cis acting sequences. The cis acting elements in general regulate genetic transcription, RNA processing, and protein synthesis. Just as in prokaryotes, genetic transcription is completed in 3 phases of eukaryote. In case of eukaryotes also. Now we are going to learn about stages of eukaryotic transcription. So they are initiation, elongation, and termination. During initiation, RNA polymerase 2 binds to the promoter site of the DNA template and initiate RNA synthesis. During elongation, the RNA transcript extends to the desired length by the incorporation of more and more ribonucleotides, and during termination, RNA synthesis stops and RNA transcript dissociates from the DNA template. Then initiation. The transcription of mRNA begins with the binding of RNA polymerase 2 with the promoter of DNA template. The promoter is located close to the transcription start site of the protein coding gene. Transcription initiation occurring in those promoters with TATA box is slightly different from that occurring in TATA less promoters. In transcription initiation, TATA box serves as a recognition site for several transcription factors. Also, it plays an important role in positioning the initiation complex exactly at the very start site with unerring precision. In this case, the first step of initiation is the recognition of the TATA box by some transcription initiating factors such as TATA binding protein, TBP, and a variety of TBP associated factors like TAFs. TBP makes sequence specific contacts with the DNA. TAF recognizes non-TATA elements and the transcription initiator of the core promoter. They also allow all transcription initiation factors to respond to transcriptionally activators and silencers. The TATA box is activated for the recognition and binding by RNA polymerase 2. So quite a number of promoters are TATs-less, yet they can initiate transcription. In them, transcription initiation is believed to be controlled by CT-rich sequence called initiator element in association with a downstream promoter element or DPE. INR is located very close to the transcription start site and DPE is located 28 to 34 pp away. The binding of RNA polymerase 2 with the promoter generates a closed promoter complex. In successful transcription, it gets converted into an open promoter complex in which the DNA remains locally unbound. Now, transcription begins with the incorporation of the first ribonucleotide mediated by RNA polymerase 2 in association with the transcription initiation factors. Soon, the enzyme moves forward from the promoter site without dissociating from the DNA transcription and at the same time freeing the promoter for further initiation. This is called promoter clearance and it marks the end of transcription initiation. Promoter clearance occurs only when the open promoter complex is stable. Usually it follows several abortive initiations in which only short transcripts are generated. Then next, elongation. Once successful initiation has been completed, RNA polymerase 2 moves forward along to DNA template, catalyzing the incorporation of more and more ribonucleotides and synthesizing a nascent mRNA in the 5' to 3' direction. This brings about elongation. During this, RNA polymerase 2 gets associated with a series of transcription elongation factors which prevents its dissociation from DNA template until it reaches the end of the protein coding gene and transcription is over, or elongation is over at least. In eukaryotes, transcription elongation is always coupled with the co-transcriptional RNA modification. During transcription elongation, the double-stranded DNA gets unwound ahead of the elongation complex and rewound behind it. This results in a transcription bubble with the disruption of the nucleosome organization of DNA. Next step, termination. The termination of eukaryotic mRNA transcription is only poorly understood. It involves the dissociation of the enzyme and elongation factors from DNA and the liberation of the nascent mRNA transcript. In eukaryotes, termination of transcription by RNA polymerase II occurs at a point beyond the end of the protein coding gene. It is believed that transcription termination probably occurs when RNA polymerase encounters either a chemical independent impediment or receives a stop signal from termination codon of the DNA template. However, no such termination signals have so far been identified. An alternative view is that termination signals are altogether absent and the dissociation of the elongation factor at some point destabilizes the transcription complex. This in turn leads to the dissociation of RNA polymerase II, termination of transcription, and the release of mRNA transcript. Next topic, which is the post-transcriptional modification or processing of eukaryotic mRNA, will be dealt with the help of next video. So thank you for listening. We have covered the genetic transcription. We just listened to a quick summary. Thank you for listening.